Welcome to E-Learning Science. I am Dr. Mudasir. Today in this video tutorial, we will be discussing types of acquired immunity. As we know, acquired immunity has other names also. It is sometimes called as specific immunity. It is sometimes called as adaptive immunity. And this type of immunity is broadly divided into two types. One is active and second is the passive. Active immunity means where our immune system is actively involved, and passive immunity means our immune system is not actively involved. For example, in case of active immunity, we can presume there is direct activation of our immune system, and that activation of immune system will lead to the production of antibodies, and these antibodies are going to kill the foreign invader who will be invading our body. This active immunity, how we are going to activate our immune system, how we are going to directly actively activate our immune system, may be natural or artificial. We may do it, we may activate our immune system in a natural way or in artificial way. How we can activate it in a natural way? It is very simple, because we can activate this immune system by directly microbes. If pathogen is coming, if there is any pathogen, if there is invader, any invader, maybe a bacteria, maybe a virus, maybe a protozoa, maybe a fungi, this invader will definitely activate our immune system. It will produce antibodies in our body, and this type of immunity is active immunity but natural. It is naturally present in the body of any organism. So this is natural active immunity. Natural active immunity can be activated, can come into the picture when there are invaders, when there is any attack of any microbe, maybe a bacteria, virus, or any pathogen. Artificial. Again, we can activate our immune system. The second way is also, we have activated immune system by way of entry of any pathogen. But there are other ways also. Our immune system may be activated artificially, deliberately, by using vaccines. When we go for immunization, active immunization, this active immunization, the rationale behind this active immunization is to evoke the production of antibodies in our body, so that the ready-made dose of antibodies against a particular disease for which vaccine has been given will remain available. In case the entry of the particular invader happens in our body, it will immediately intensify the attack against them and will neutralize them. Again, our immune system is activated in this stage also. Here it is naturally getting activated, but here it is activated artificially. Discussing these vaccines, I must tell you one thing. A pathogen, any pathogen, let's discuss this part. This is done. Artificial active immunity and vaccines. Remember, a pathogen has two properties. Any pathogen has two properties. One property, the first property of pathogen is antigenicity. And the second property of pathogen is pathogenicity. Pathogen is any foreign invader or any microbe. The pathogen has two properties. One property is antigenicity and second property is pathogenicity. Antigenicity is the property of antigen by which it can evoke our immune response. It can evoke our immune response. It is the natural tendency of our body. Whenever we are getting any foreign invader, whenever our body is being attacked, at that time our body has a natural tendency to fight against the invader, because this invader, it is the inherent property of this invader, this pathogen, to have antigenicity, to possess antigenicity, and by virtue of this antigenicity, our immune system gets activated and it will evoke immune response against the pathogen. This is the property of pathogen. Second, pathogenicity. It is not only whenever any pathogen is invading our body, it is not going to activate the immune system only, but at the same time it gives us harm in a number of ways. And this property of pathogen, the property of pathogen by which it gives harm to the organism, to the man, to the person, is the pathogenicity. Every disease is associated with signs and symptoms. There are a number of symptoms which are related to a particular disease. So these symptoms will be caused. These symptoms are caused by virtue of one property of the antigen, that is pathogenicity. Right? See, when we look towards the rationale of vaccines, vaccine preparations, in all type of vaccines, our basic aim and ambition is to eliminate the property of pathogenicity and to retain the property of antigenicity. We are giving a vaccine, and that vaccine may involve the organism directly who is going to do the disease. But we are not getting sick. What is the reason? The reason is that this is the same organism probably, or as some part of that organism, but this organism has only one property, that is antigenicity, but not pathogenicity. We are eliminating the property of pathogenicity in case of pathogen, and we are retaining the property of antigenicity, by which it will be able to evoke our immune response. And by evoking immune response, our immune system is activated. We will be having B cells, we will be having memory cells, and these memory cells will remain. For example, if next time the active invader will come to our body, definitely our body is going to have a prompt response against the pathogen. And these vaccines are basically of 4 types. Let's have a little bit discussion of vaccines. One vaccine is, we call it as inactivated vaccines. We have second category, live attenuated vaccines. We have one more category, toxide vaccines. One more, subunit vaccines. Broadly, vaccines are categorized into 4 types. We may have inactivated vaccines, we may have live attenuated vaccines, we may have toxide vaccines, we may have subunit vaccines. When we discuss this inactivated vaccine, in this case, we are preparing the vaccine by using the organism which is going to cause disease, but this organism is dead. It is dead. It may have the property of antigenicity, but should definitely lack the property of pathogenicity. We are using the dead microbes, and these dead microorganisms are being given maybe in IV route, maybe in intramuscular route or subcutaneous route, whatever route is available. And this vaccine is going to evoke immune response. But such kind of vaccines, since we are using dead organism, they oftenly require booster doses. Booster doses are desired. Booster doses are desired in such kind of vaccines. And we have flu vaccine, we have hepatitis A vaccine, we have cholera vaccine. Such kind of vaccines are basically inactivated vaccines. Then we have live attenuated. Here, the microorganism we are using live. It's a live microorganism, not a dead microorganism in this case, as it was in this case. But we are using a live microorganism, attenuated, heat killed. It has been attenuated. it has been we can say the powers of this antigen, the powers of this microbe has considerably been brought down. And this type of vaccine must have antigenicity, but should lack what? Pathogenicity. There should be antigenicity and there should not be any kind of pathogenicity. These vaccines are usually, they do not require booster dose. They do not require frequent booster doses. Frequent booster doses are not required. They can produce the immune response that will last for much longer period as compared to inactivated vaccines. Then the third type of, in this case we have measles, mumps and rubella vaccine, MMR. Measles, mumps and rubella vaccine is one of the example of live attenuated vaccines. Then we have toxoid vaccines. Toxoid vaccines are basically made up of a toxin, but the toxin should be inactivated toxin. For example, we have tetanus, we have diphtheria. The tetanus and diphtheria microorganisms are infecting a person and they are causing the pathogenicity, they are causing the damage to the organism by producing the toxins, by their toxins. And what we are doing in such kind of vaccines, we are preparing the toxoid vaccines. We get that toxin, but that toxin should be inactivated to a considerable level until we can say it should have the antigenicity, but it should lack the property of pathogenicity. These toxins are directly used, and these toxins will directly activate our immune system, and they can give us respite for a particular period of time from procuring such type of disease. And toxoid vaccines, we have tetanus vaccine, or we have diphtheria vaccine. Tetanus vaccine or diphtheria vaccine. Then we have one more category, that is subunit vaccine. Subunit vaccines are composed of small fragments of organism. Small fragments. Of organism. Here, we may be using a complete organism. We may also be using a complete organism. We may be using a toxin of a particular microbe. But in this case, we are only using a small fragment of, a small portion of that particular organism. We do not require a complete organism. We require a particular small fraction of that organism. This type of vaccination was possible by way of recombinant DNA technology. These vaccines were made by way of recombinant DNA technology, and the example of this type of vaccine is hepatitis B vaccine. So these are the different categories of vaccines. Anyways, we were discussing this thing. All the vaccines, whatever we have discussed, are basically making a part of artificial active immunity. We are giving them artificially, and they will evoke immune response, and that too active immunity, because they activate the immune system directly. Right. Then we have the second type of immunity. After discussing this active immunity, we have the second type of immunity, which we call as passive immunity. Passive immunity involves the direct use of we can say antibodies. We are directly giving the antibodies. In this case, we are not making sure to activate the immune system of person. Immune system of person is not going to get activated. What we do in this case there may be transfer of antibodies, and that transfer may be in a natural way or that may be in artificial way. Now imagine the case where we are getting the antibodies, we are receiving, any person is receiving a dose of antibodies, and that too in a natural way. Yes, it is going to happen in case of mother-fetus connections. If we see there is transfer of mother, in case of mother to fetus, we see there is transfer of antibodies like IgG from mother to fetus through placenta, when the fetus is in the womb. So these IgG is a class of antibody, and antibodies are present in the mother, and this type of antibody is formed by the mother, and this type of antibody is transported to the fetus through placenta, and this fetus will receive these antibodies naturally and is not going to activate its immune system, but definitely what it will do, it will prevent the fetus from getting any kind of disease, and therefore giving him a type of immunity which we call as passive immunity. But this passive immunity is natural passive immunity, and this is being achieved by the transfer of antibodies from mother to fetus. One more thing, again there is the same connection, transfer of antibodies from mother to fetus. It also takes place, this transfer of antibodies takes place at the time of breastfeeding. The milk of mother has antibody IgA, and this antibody is given to the, not fetus, it is given to the child. It is mother-child connection. It is given to the child, and this child receives a dose of antibodies directly through the milk of mother, and these antibodies are going to protect the child against infections, and without getting activation of any child's immune system, he is receiving a direct ready-made dose of antibodies, and these antibodies he is using for combating against the various infections, providing him a type of natural passive immunity. And the last is artificial passive immunity. Again we are giving antibodies, but these antibodies are being given artificially. We are providing antibodies. For example, ADS. What is ATS? Anti-tetanus serum. ADS is anti-diphtheria serum. Anti-tetanus serum is a ready-made dose of antibodies. What it contains, it is a ready-made dose of antibodies which are given to a person artificially as a preventive measure, to protect him from getting the infection of tetanus. For example, how we prepare these ATSs? ATS anti-tetanus serum is being prepared by giving a direct immunization of tetanus toxide to the horse. We are injecting the horse by a tetanus toxide, and what is going to happen? The horse, this tetanus toxide, it has antigenicity but lacks pathogenicity. The horse will produce antibodies against it, and these antibodies are present in the serum of horse. After some days, what we do, we take the blood from the horse, we extract the serum, and we extract the antibodies against the tetanus in the serum, and these antibodies we call as anti-tetanus serum, which we are given to human beings whenever they have a deep injury, or whenever we feel there may be a threat that this tetanus, the person may encounter any kind of tetanus organism at any point of time. As a preventive measure, we advise them to take ATS, or sometimes we also advise to take ADS whenever it is required. So this is the type of acquired immunity. I hope you must have understood, and if you like, or you want to share, or you want to comment on the video, or if you want to demand any kind of lecture in the future, I will definitely appreciate the move. Thank you very much.